Okay, thank you, Myron. I'm delighted to be here with you this morning. I want to thank Myron and the organizers for the invitation. I have to admit that when Myron invited me, I said, Myron, you know I'm not a physicist, right? And you know I don't do physics, right? And so I'm going to confess that I'm not going to talk about physics today in Saturday morning physics. But hopefully I still have an interesting scientific story to tell you. I call myself a geomicrobiologist. And that means that I study how tiny organisms, like bacteria, have shaped our planet, how they've done that through the planet's history and how they continue to do that today, and also how they shape the environment around us. And most of my career, I have studied deep-sea hydrothermal vent systems. All right? These are hot springs on the seafloor, and I ask questions about how are these microorganisms living off of the chemicals that come from deep-sea hot springs, and how are these microorganisms affecting the chemistry of the oceans. But today, I want to focus on a new passion of mine, my new love, which are these mighty cyanobacteria. These are tiny organisms that have literally transformed our planet. Okay? And there are a couple things that have gotten me into these organisms in the past couple of years. The first is that I teach an undergraduate geology class. I teach undergraduate geology majors about the coevolution, the changing of Earth and life together through time. And when we think about how biology has impacted this planet, at the very top of this list, right alongside humans, are these organisms of cyanobacteria. And that's because these are the organisms that invented oxygen-producing photosynthesis. Okay, so these guys have invented this metabolism and literally breathed oxygen into our atmosphere through time. The second thing that got me into these organisms, soon after I arrived at Michigan as a young assistant professor, this was about 8 years ago now, I was looking for deep-sea vent analogs in the Great Lakes. So we were exploring the Great Lakes, looking for deep-sea vent analogs, and we found them. I'm not going to talk about that today. But we also stumbled upon these amazing cyanobacterial mat systems here. These are mats of cyanobacteria that we think resemble what the early Earth looked like for billions of years. So this provides us with a window into the early Earth. It allows us to study biology as it existed for these long stretches of Earth history. And then the third thing that got me into these cyanobacteria is represented in this picture on the right. This is a picture that we took last summer on our boat in Lake Erie. And what you're seeing is this pea soup colored water, that's Lake Erie water, that has very dense concentrations of cyanobacteria. So this is an example of cyanobacteria as troublemakers. Okay? These are organisms that threaten drinking water supplies worldwide, from Lake Erie to Asia to Europe and South America. This is a global threat to drinking water systems. And so this is the type of problem that my mom understands and is happy to tell her friends about, right? It affects our daily lives, and we're trying to understand this problem so we can understand how to keep our drinking water supplies available and in high quality. Okay, so first I'm going to tell you about both of these stories today, but first we're going to start with Earth's oxygenation, and actually want to begin with a little audience participation exercise. Don't worry, this is going to be easy. I'm going to ask you to breathe, to take some deep breaths in and out. Okay, so take some deep breaths. Enjoy that oxygen that you're breathing. It turns out that we do this about 20,000 times per day, right? We do this 20,000 times per day, and it's not just us, it's all other animals on this planet that are using oxygen, and in fact, many bacteria also breathe oxygen. And collectively, it turns out we are using a substantial portion of oxygen. Biology is, okay? We're consuming this oxygen. We breathe that oxygen into our lungs, right? Think about that as you continue to breathe. That oxygen is transferred to our blood supply and delivered to tissues, and that oxygen then provides energy when it reacts with sugars, right, and produces ATP, which we use to move and to grow and to think, right? So the key here is that oxygen is being consumed, and so you and I and all other animals together, we're participating in what we think of as the global oxygen cycle. We are consuming that oxygen. Okay, so congratulations, you've participated in the global oxygen cycle this morning. I said this oxygen is being consumed, and if it wasn't for a constant supply, a constant resupply of this oxygen, it would be consumed over relatively short time scales, on the time scale of about 4,000 years. That doesn't sound short, but on geologic scales, that's really short, okay? So we need a constant resupply of this oxygen. And luckily, we have it, right? This is a textbook example of photosynthesis. And so what happens in photosynthesis? We have organisms like these plants here, which are using sunlight to convert carbon dioxide into sugars, and that's where our food comes from. But they're also extracting electrons from water. They're oxidizing water, and in the process, they're transforming that H2O into O2, right? That oxygen from H2O is converted into O2. So this is where our oxygen comes from. And that O2 and that sugar then is recycled. It's used by organisms like you and I that are conducting respiration. So if you look at these two equations, respiration is essentially the opposite, the reverse process of photosynthesis. Okay? So in our modern world, we have this tightly coupled photosynthesis and respiration. And thanks to this tight coupling, atmospheric oxygen concentrations don't change very much. Okay? So luckily we have constant oxygen concentrations. But that has not always been the case. If we look back through Earth history, so here now I'm showing a plot of atmospheric oxygen concentrations. That's shown on the y-axis in terms of percent of current levels, right? So from 0% to 100%. And on the x-axis, we're looking at time in terms of billions of years ago, from 4 billion years ago to the present at 0 billion years ago. So we can see that, in fact, oxygen concentrations in the atmosphere have varied tremendously. Okay, if we start here in the present, I said these oxygen concentrations are relatively consistent these days, and I'm simplifying this. There are some wiggles here that I'm not worrying about at the moment. But if we go back about 500 million years, 500 million years, we see this big step in oxygen concentrations that occurred. Okay, before that, these oxygen concentrations were very low. They were between about 1 and 10% of current levels. And that was the case for about 2 billion years. Okay, for most of Earth's history, oxygen was low. And if we go back even farther, back to this, what we call the Great Oxidation Event, this is another big step in oxygen concentration. Oxygen was essentially not present in substantial quantities. Okay, so for much of the early history of this planet, there was very little oxygen, not enough oxygen to be useful to biology. All right? So we're talking about billions of years here. We humans are not used to thinking about billion-year timescale. So I want to give you some markers here to think about this timescale. Humans have been around for something like a few million years, if you consider our relatives as well. Okay, so we are just the latest blip in the history of life on this planet. If we think about some ancient organisms that are now extinct, like dinosaurs, right? They were around from about 60 million years ago to, let's say, 250 million years ago. So they're right in here. Again, relatively recent in the history of our planet. You may have heard about the Cambrian explosion. This is when animal life diversified, right? That was about 500 million years ago. 542 million years ago was the Cambrian explosion. And it's probably no coincidence that this explosion, this radiation of animals, occurred at this step up in oxygen concentrations. Large animals like us need lots of oxygen to move and to respire and to be active, okay? And to give you a sense of what oxygen levels were like in this middle period of Earth's history, 1 to 10%, how does that compare to the top of Mount Everest? Well, the top of Mount Everest is about 31% of the oxygen levels that we have here. So this was very low oxygen, not enough oxygen to support people and most large animals. Okay? It used to be that back in Darwin's time, we thought of this Cambrian explosion as the origin of life itself, because this is when, in the rock record, we see this appearance of fossils. Okay? But it turns out that the history of life goes way back before that, way back until something like 4 billion years ago. So there's this long stretch of Earth's history before animals that was dominated by microorganisms. This microbial world dominated life on our planet. Darwin was actually worried about this. He called it Darwin's lost world because he was concerned that if life originated here at 500 million years ago, there wasn't enough time for his evolutionary process to give us the diversity of organisms that we see. So in fact, this was not the beginning. We go way back to 4 billion years ago. And here now I've added our current best knowledge about the origins of these mighty cyanobacteria that I'm introducing to you. So we have evidence that cyanobacteria were present at about 3 billion years ago. And this is interesting because there's a long period of time between the origins of cyanobacteria and this great oxidation event, something like 600 million to 1 billion years, right? So if oxygen-producing cyanobacteria were present, why did it take so long for oxygen to accumulate? That's one big question. Another big question that we don't understand is that why did oxygen remain so low for so long, for 2 billion years? These big questions are key to understanding the evolution of life on our planet, right? The development of animal life. Ironically, we don't know very much about how cyanobacteria live under these low oxygen conditions. These low oxygen conditions that were pervasive throughout their evolutionary history. So my lab is focused on trying to understand how these cyanobacteria work under low oxygen conditions. And one of the main questions that we're addressing is, can these organisms produce oxygen in the absence of oxygen? Can they produce oxygen under the types of low oxygen conditions that existed during this great oxidation event? All right? Now, unfortunately, we don't have a time machine to go back and visit these ancient microbial communities. We're waiting for the physicists to figure that out for us. We would very much like to go back in time. In the meantime, we have to settle for a couple of different ways of studying these communities. Number one, we can look back at the geologic record, and we can look at the signs of life that lived in those periods. And when we do that, indeed, we see that the world was covered with things like stromatolites, fossilized microbial mats. And that's how we know that microbial mats were very abundant and very pervasive during the early Earth. But the other way that we can study such processes is by looking at modern living systems that resemble past life. Okay, so we're looking at modern analogs of ancient microbial ecosystems. And one of the very best analogs of ancient microbial ecosystems occurs right in our own backyard here in Lake Huron. So here I'm showing you a cyanobacterial mat. This is think of it as like a carpet of cyanobacteria that's growing on the bottom of Lake Huron in about 23 meters of water. This is just up near Alpena, Michigan, for those that know northeastern Michigan. So I'd like to introduce you to this modern analog of ancient ecosystems. To understand this, we need to understand something about the geology of Michigan. Okay, so you might know that Michigan is a basin. It's like a bowl, right? And this basin once was filled with seawater, with saltwater oceans. So where we're sitting now was an ocean about 400 million years ago. And those oceans deposited sedimentary layers of rock. If you look at this bottom here, this is now a cross-section of Michigan with Lake Michigan on the left, Lake Huron on the right. So we're looking at Michigan from the south. And we have this cross-section with these different layers of sedimentary rock. And those layers form these concentric rings here of different sedimentary rocks. Okay? So the key here for understanding our modern analog of an ancient system is that as those oceans in some periods dried up, they left behind deposits of salt, right? As the water evaporated, salt was left behind. We call these deposits evaporites, and we mine these deposits for salt. And one of the important salts in those evaporites is sulfur, sulfate, gypsum. You might have heard of gypsum, right? Calcium sulfate. So what happens in these systems is we have groundwater from, let's say, rain. Rain falls onto Michigan. It seeps into the groundwater. It interacts with these evaporites, and it dissolves the salt. Okay, so now we have groundwater that's salty. In that very same groundwater, we have organisms, bacteria, that are respiring. They're doing that breathing that you and I are doing, and they're consuming the oxygen. Okay, so we end up with salty, sulfur-rich, oxygen-free groundwater. And that groundwater circulates through these subsurface here, and in some cases, it finds its way back to the surface in groundwater seeps and vents. Okay, so it reemerges as vents. And one of the places where this happens is in sinkhole systems. So these are sinkholes in northern Michigan where the rock layers, the limestone has dissolved and formed a sinkhole, and into that sinkhole groundwater is percolating up. Okay, so we have these groundwater vents that are coming up into sinkholes. And the example that I'm going to tell you about is this island called Middle Island, not far off the coast of Alpena. And from this aerial photo that we took from a plane, you can just make out the sinkhole that I study. This is called the Middle Island Sinkhole. All right, if we zoom in a little bit closer, you can see the outline of this sinkhole. So again, this is where the limestone has dissolved, and we have groundwater that's bubbling up into the gap, the cracks in the rocks here. Here's a boat for scale to give you a sense of how large the sinkhole is. It extends all the way out here, and it's several football fields in size. Here's now a cross-section of the sinkhole that we study. So we have this water, this groundwater that's coming up from sediments. It's bubbling up into this sinkhole. So we're looking at it again from the side in cross-section. This salty water is more dense than the lake water. Okay, so it sticks to the bottom, and it fills up this bowl, something we call the alcove here, with salty oxygen-free water. And then that salty oxygen-free water, because it's heavy, it fills up the bowl, and then it spills over this sill and fills this larger sinkhole area to the right here that we call the arena. Okay? Divers describe this as an exciting sort of underwater waterfall that they can ride as they go down the sill, because it's this underwater, salty, dense waterfall, right? Okay, key to us is that, again, this water contains undetectable oxygen, very low oxygen concentrations. And because of those low oxygen concentrations, it excludes regular lake life. Okay, there are no fish in this system. Animals like you and I need this oxygen. So in the absence of regular lake life, these cyanobacterial mats, these beautiful purple carpets of cyanobacteria are able to thrive, just like they did before the evolution of animals and plants. So here we have this sort of pre-Cambrian, pre-animal and plant ecosystem. This is hard to describe in words or with still pictures, so I want to play you a quick video here. What you're going to see is a remotely operated vehicle, a little robot that we've put on the bottom of the lake that's zooming over these microbial mats. Everything that you're going to see, all the colors, the white and the purple, is microbial. And we've set it to some otherworldly music here about the early Earth. Typically, I have to travel to the bottom of the ocean, or at least the Yellowstone National Park, to see such extreme environments. It's a fantastic discovery to find these in our own backyard here. When I dream about the early Earth, this is what it looks like. Early life. Okay, if we zoom in to those microbial communities now, you can start to see these beasts. These are essentially miniature versions. You can think of these as filamentous, sort of worm-shaped organisms on millimeter scales. These are some of the most social bacteria that I know of. If you take these mats and you disperse them, you break them up from their friends, they're mobile. They will crawl back together and reform these structures and rebuild their cities. Okay, so these are social organisms. They like to be together. They cooperate. Occasionally they antagonize each other and they fight with each other. But for our purposes, we're mainly interested in this process of oxygen production, right? So we have cyanobacteria that are thriving in a low oxygen environment. And the question is, do these organisms produce oxygen in the absence of oxygen? And so to address that question, we started out with very simple experiments where we deploy what we call these benthic chambers, benthic metabolism chambers. These are just plexiglass chambers that are gas tight that have a sonde here that measures oxygen concentrations. Okay, so we're measuring oxygen concentrations just above these cyanobacterial mats in the light, shown on the right here, and on the left here, this is in the dark. So the expectation is, if we're seeing oxygenic photosynthesis, is that we would see light-dependent oxygen production, right? We would see light-dependent oxygen production. Here's the data. Do these cyanobacterial mats produce oxygen? Here I'm showing you the concentration of dissolved oxygen on the y-axis as a function of time. So we're looking over the course of several days, a couple of day-night cycles here. And this is showing both in the dark and in the light, in uncovered systems where we have a day-night cycle, okay? And what I want to point out is that in both cases, we actually see a net decrease in oxygen concentrations. This was stunning to us. We think of these cyanobacteria, they're famous for producing oxygen, but in this case, they appear to be net consumers of oxygen, right? So how could these cyanobacteria that do oxygenic photosynthesis be consuming oxygen? Well, one hypothesis is that there are forms of photosynthesis that do not produce oxygen. Okay? And I'm going to introduce you to this form of photosynthesis by comparison to the photosynthesis that you all know and love, oxygenic photosynthesis. All right? So oxygenic photosynthesis is the one that I introduced you to before. Remember, we're using carbon dioxide and water, and we're converting that with light to these sugars and to oxygen, right? And I told you that the oxygen actually comes from the O in H2O, right? So oxygen is being produced because we're using H2O. Well, in anoxygenic photosynthesis, instead of using H2O, we are using H2S, okay? And when organisms use H2S, the rest of this is the same. They're using CO2, they're using light, but when they use H2S, the product, instead of O2, is sulfur, elemental sulfur, globules of yellow sulfur. Okay, so this is a form of photosynthesis called anoxygenic photosynthesis. It is done by these organisms called purple and green sulfur bacteria, but there's evidence from us and from other groups that these cyanobacteria can also do anoxygenic photosynthesis. Okay, so these are potentially versatile organisms. So to test this hypothesis, actually before I get to that, I'll say, so, is there a source of sulfur in these systems? Yes, I introduced you to that sulfur that's coming from gypsum. And in fact, when we do chemistry in these systems, we see that there is plenty of this hydrogen sulfide around. There's plenty of the H2S. That's what's being shown here in this red curve. This is the concentration of sulfide, or hydrogen sulfide. If you smell rotten eggs, right, you know that stinky hydrogen sulfide smell. That's what this is here. And it's being produced by organisms called sulfate-reducing bacteria. These are organisms that are breathing sulfate and exhaling sulfide. Okay, so they're producing the sulfide, and they're leading to this high concentration. As this red line here moves to the right, that's indicating a high concentration of hydrogen sulfide. Okay, so there's good reason to suspect that these cyanobacteria here are utilizing this H2S for anoxygenic photosynthesis. Okay, so could this be happening? To answer this question, we collect these cyanobacterial mats. So shown on the left is a push core. We take a core of these sediments and take the cyanobacteria along with the underlying mud, and we bring them back to the laboratory where we can incubate them under simulated environmental conditions. Okay, the same temperature and the same light levels that they experience at the bottom of Lake Huron. And as we have them in the lab now, we can torture them and we can ask them questions. We can say, what type of photosynthesis are you doing? We can do experiments on them and we can study them. And one of the first experiments that we did is we measured rates of what we call primary production, rates of fixation of carbon from carbon dioxide into those sugars, right? This can include photosynthesis as well as what we call chemosynthesis, synthesis that's driven by chemistry like sulfur eating, okay? And we've done these experiments under two different conditions. On the left, we've kept these purple cyanobacterial mats intact with their underlying sediments. And the importance here is that, remember, the sediments are what's producing that smelly hydrogen sulfide gas. Okay, so here we have cyanobacterial mats as they exist in nature, in contact with that hydrogen sulfide. On the right, in this experimental condition, we've removed those cyanobacterial mats from that sedimentary supply of sulfide. Okay, so now we have cyanobacteria under oxygenated conditions without hydrogen sulfide. And under these conditions, we feed them carbon-14 labeled carbon dioxide. So we're feeding them their carbon source, and we're measuring how fast these organisms do photosynthesis or chemosynthesis over time. Okay? And here are the results. So on the left, I'm showing the results of carbon fixation over time in the case where the cyanobacterial mats are in contact with their underlying sediments. And when there's enough sulfide around, when they have that sulfide supply, you can see that these organisms do not produce oxygen. They're conducting anoxygenic photosynthesis and also chemosynthesis. Again, this is through eating the sulfur without using the sunlight. In contrast, if you look to the right now, we're looking at the amount of carbon fixed per area per day. And in the absence of the underlying sediments, so here we have cyanobacterial mats without the sulfide, these organisms switch over to oxygenic photosynthesis. All right? So this is consistent with the idea that these cyanobacteria indeed can conduct both types of photosynthesis. The modern form that you know and love, the oxygen-producing form, but also the ancient form that uses hydrogen sulfide. Okay, so we thought we had it figured out. We published this in a paper in 2012. This is a graduate student that did this work. Until later that year, we did our first dives on the system at night. And when we dove on these mats at night in the dark, we were stunned to see that our old friends, the purple mats, seemed to have left and been replaced by these white mats on the right, a totally different appearance for these microbial mats. You can see some remnants of these purple guys here, okay, so they're still there. Okay, so what's going on here? Well, we theorized that what's happened is we've gone from this situation on the left, where we had purple cyanobacteria on top and white sulfur-eating bacteria on the bottom, to a new state where the white sulfur-eating bacteria have moved to the surface. And there's good reason to expect that this has happened. And that's because the concentrations of sulfur change on a daily cycle. Okay, the sulfur, which again is shown in red here, higher concentrations on the right, that sulfur concentration is migrating upwards. Okay, so when no oxygen is produced, that allows the sulfide to move up in the sediments and up into the microbial mats. So what we think we're seeing here is a microbial migration, right? Microorganisms, the white guys here, are moving to follow this supply of sulfide upwards. So to test this, we again brought these microorganisms back into the laboratory, and I have a brand new postdoc in my lab. This is Judith Klatt, who's just moved here. She came all the way from Germany to study Lake Huron. And Judith is an expert in the use of what we call microsensors. These are chemical sensors that can be used to detect and measure concentrations of hydrogen sulfide and oxygen at very fine spatial scales. We can measure these chemicals at 10 micron spatial scales, 10 millionths of a meter. Okay, so we can measure these chemicals at very fine spatial scales. And we can measure these profiles that I've introduced you to. And if we're clever, we can also use these tools to quantify the rates of the different types of photosynthesis, oxygenic and anoxygenic. We do that by doing experiments in the light and in the dark, and also by using inhibitors of oxygenic photosynthesis. So this is data that was just produced last month. You guys are some of the first to see this data. It's unpublished. And what I'm going to show you are these chemical profiles on the left, and also our measured rates of anoxygenic and oxygenic photosynthesis on the right. Okay? So we'll start in the dark without any lights on. And in the dark, as we would expect, there's no photosynthesis taking place. And as we hit the lights, especially at low levels, simulating light levels that you would see in the morning, we start to see the consumption of sulfide. Okay, so this red line moves to the left. Did everyone see that move to the left? So from dark to light, it moves to the left. That means sulfide is getting consumed. And based on that consumption, we can measure rates of this non-oxygen producing photosynthesis, this light-dependent sulfide consumption. Okay? If we turn the lights up a little bit higher, like we would have at midday, we see more photosynthesis. We see faster sulfide consumption, and we can quantify and estimate these rates of anoxygenic photosynthesis. All right? And if we let this go longer, what we see eventually is that this concentration of sulfide continues to be depleted to the point where it's completely depleted. Okay? And this is the time point where we start to see anoxygenic photosynthesis petering out, right? It's lost its supply of H2S, and oxygenic photosynthesis starts to kick in. Okay, so that's going to be shown here on the right in blue. This is also the time point where we have actually now observed in the laboratory this microbial migration. Okay, so indeed we see the white sulfur-eating bacteria start to go back down into the sediments. They're following their food source, their sulfur supply, and the purple cyanobacteria now are coming up to take advantage of that light. Okay, so we see this microbial migration happening on timescales of hours. If we continue to let this go, again, these organisms don't have H2S, so they've now fully switched over to using H2O as their source of electrons for photosynthesis. So now they're really going gangbusters, producing oxygen, and we don't see any anoxygenic photosynthesis. So the lessons learned here are that these cyanobacteria can do both oxygenic and anoxygenic photosynthesis, but it very much matters the time of day that you're studying them. You have to study these guys on the timescales that they care about, the timescales of hours. Okay? Okay, so far I've been telling you about how we measure the end products of these microorganisms, their metabolisms. And now I want to transition a little bit and tell you about how we study the organisms themselves. How do we identify which species are present? How do we identify what metabolisms they are capable of? How do we study organisms that we can't see with the naked eye? And I give my senior colleagues a hard time because for years they've been using these rather low-tech strategies for studying microorganisms, right? Here is my good friend and collaborator Bopi Bananda from Grand Valley State. He's looking into the water with his snorkeling mask on. And in fact, in our case, you can see these microbial mats all the way at the bottom of Lake Huron. The water quality is fantastic in Lake Huron thanks to the zebra mussels that have clarified it for us. Okay? But still, visually, we can't study these guys in great detail. So how else can we study microorganisms? Well, one way to study these microorganisms is to culture them. Very much like you would cultivate a plant, you can cultivate bacteria. This is what we've done since the days of Pasteur, right, for over 100 years. This is fantastic. Again, we can bring these organisms into the lab, we can interrogate them and study them. This is great. The downside is that most organisms in nature, most organisms out there in the diag soil, or even on the surface of your skin, refuse to grow in culture. They refuse to come into captivity. Something like 99.9% of all organisms have never been brought into culture. When we first realized this, it was an oh no moment. We've been missing most of the microbial world. Okay? You might also think that as microbiologists, we can use microscopes to study these organisms, and that is absolutely true. There's all sorts of powerful microscopy techniques, and they have their strengths and weaknesses. One of the weaknesses is that many organisms that are completely different, they're different phyla, look exactly the same under a microscope. Okay, so microscopic observations of these microbes have their limitations. So what my lab really focuses on, and what a lot of microbiologists now do, is instead of growing these organisms, instead of looking at these organisms, we take samples of the environment, be it water or soil or microbial mat, we extract DNA from those samples, and we sequence that DNA to know the organisms that are present through their genes. Okay, so we think of these genes as markers of which organisms are present and what their metabolisms and functions can do. What are their roles in the ecosystem? So here's a schematic of what we call environmental genomics. So here we are studying organisms that have never been brought into culture. Okay? And so we might start with a sample here, like a cyanobacterial mat. We extract the DNA. We also do this with RNA and protein. We end up with DNA from tens or hundreds or maybe even thousands of different organisms. Okay, so we then have to fragment that DNA into many little pieces in order to sequence it. We do what's called shotgun sequencing, which means we sequence all of it. We're not picky about what we sequence all of it on these fancy new high-tech DNA sequencers. And these days, we can, for a relatively low cost, produce billions of DNA sequences. The declining cost of DNA sequencing is staggering. 15 years ago, it cost something like $1 billion to sequence a human genome. We're now approaching a $1,000 human genome. Okay, this is one of the few technologies that's moving much faster than computer processing speed, advances in computer processors. When I was a graduate student, I was happy to produce hundreds of sequences. My graduate students now produce billions of sequences at a time, and it's not because they're working thousands of times harder than I did. It's because technology's come a long ways. Okay? So we then end up with billions of DNA sequences from perhaps thousands of different organisms, and we're confronted with what I think of as the world's largest puzzle, right? We need to then put those DNA sequences back together and assign them to different organisms. And luckily we have computer algorithms that allow us to do this, and we piece these DNA sequences back together, back into their whole genomes, the DNA that encodes individual organisms. And now we have blueprints of these organisms that we've never had in culture. So from these blueprints, we can understand what these organisms do and how they interact with their environment. I'm going to spare you the details of this study and just give you the summary. Okay? Again, this is work done by graduate student Alex Voorhees, and using these environmental genomic techniques, he found that these mats are indeed dominated by cyanobacteria, and that these cyanobacteria can indeed do both the oxygen-producing and non-oxygen-producing forms of photosynthesis. Another major finding was that these cyanobacteria do not produce one of the main compounds, this is a lipid, that we use to detect these organisms in the geologic record. Okay? This highlighted to us that we do not have good tools of looking at these organisms through geological time. Okay? So there's a gap in our knowledge in studying these organisms. And then the final major conclusion here was that these cyanobacteria are really novel. These organisms are much different than what we've studied before. They're here, right here in the Midwest, and yet they're quite novel organisms. This work was published in 2012. Okay, so to summarize what I've told you so far, I've introduced you to these Great Lakes sinkhole ecosystems. These are analogs, we think some of the best analogs on our planet, of ancient ecosystems that once dominated the Earth. These cyanobacteria are somewhat unique in that they can conduct both types of photosynthesis. They can do oxygen-producing or non-oxygen-producing photosynthesis. And I think of this as a really important decision that these organisms make. And thank goodness they made the decision over time to produce oxygen for us, right? But we're now very interested in studying this regulation of oxygen-producing and non-oxygen-producing photosynthesis. We now know that not only is sulfide key in this decision-making process, but the availability of light is also critical. This is something that we hadn't thought about before in determining the regulation of these two different forms of photosynthesis. So it's not just chemistry, it's also light that matters. So we're now trying to map light availability back onto the early Earth and onto different habitats to think about how that would affect oxygen production. Okay, and the last slide that I'll show for this part one is this pattern of Earth's oxygenation that I introduced you to. Okay, so if we go back and think about why did it take so long to get to the Great Oxidation Event, we now think that this anoxygenic form of photosynthesis might have slowed things down. So long as there were reducing chemicals like hydrogen sulfide present, the cyanobacteria may have preferred the non-oxygen producing form. Okay, so this may have delayed the Great Oxidation Event. It may have delayed the second rise in oxygen for this middle 2 billion years of Earth's history. And so this could be really important in understanding the evolution of animal and plant life on our planet. Okay, with that, I'm going to now switch Great Lakes. We're going to move from Lake Huron down to Lake Erie. And this is also a good opportunity to remind you what an amazing resource these Great Lakes are. These Great Lakes hold 80% of North America's surface fresh water, 20% of the world's surface fresh water. You are surrounded by arguably one of the Earth's greatest resources right here in the Great Lakes region. I'm going to talk about Lake Erie. You can see even from this satellite photo that Lake Erie looks different than the other lakes. It's shallower. And in fact, here we're seeing sediments that have been kicked up into the water column of Lake Erie. Okay, so it's shallower. It is also much more heavily impacted by people. We have Detroit, we have Toledo, we have Cleveland. This is a major population center right around Lake Erie. So this exerts pressure on Lake Erie. We're dumping fertilizers into Lake Erie in the form of sewage and in the form of farm runoff. We're also using Lake Erie. Tens of millions of people use Lake Erie as a source of drinking water. Okay, so Lake Erie is heavily impacted. One of the main issues has to do with agriculture. Okay, so here now I'm zooming into Lake Erie and I'm showing you a satellite view of this large cyanobacterial bloom. The green color that you see there is a large cyanobacterial bloom. And what's causing this cyanobacterial bloom are inputs of nutrients from these rivers. And the main culprit here is the Maumee River. Okay, this delivers something like 80% of the nutrients to Lake Erie. And most of these nutrients are coming from runoff from farms. Okay, and most of that is from fertilizers that are being applied to sustain high productivity crops. Okay, so we have inputs of nutrients that are intended to fertilize crops, but in this case, they're actually fertilizing Lake Erie. Okay, so we're seeing this massive cyanobacterial bloom in Lake Erie. If you zoom in to that, this is now that picture from our boat that I showed you. It's an amazingly dense concentration of cyanobacteria. These things can smell and they don't look very beautiful, but actually it's not a huge problem, except for the fact that these organisms, some of these organisms, also produce deadly toxins. Okay, so this was made famous in 2014 when this harmful algal bloom moved unusually right into the vicinity of the Toledo drinking water supply intake pipes. Okay, so this is actually the Toledo drinking water intake, and here is a unconcentrated sample of Lake Erie water. So we had this really dense cyanobacterial bloom right at the drinking water intake of Lake Erie. And I mentioned that the main problem with these particular cyanobacterial blooms is that they produce this toxin, this compound that's shown right here. It's called microcystin. That is the new name for this toxin. When it was originally discovered, it was actually called fast death factor. And it was called fast death factor because it's an extremely potent toxin. It kills, at high concentrations, it really effectively kills animals, okay, in laboratory studies. So this is a lethal, potentially lethal toxin. At lower concentrations like the ones we find in the lake, it can also potentially act as a carcinogen. So it's a really great concern when we're getting our drinking water from this area. And so in that week of August of last year, 2014, this led to a 3-day shutdown of Toledo's drinking water supply. The National Guard was called. There was water being delivered from other cities. This was a major public catastrophe. So we pretty much know where the fertilizers are coming from. Okay? We pretty much know what's causing these blooms. But one of the things we don't understand is what controls the toxicity. And this is an interesting question because this organism, this species of cyanobacteria, Microcystis aeruginosa, has strains that can either be toxic or that can be non-toxic. Okay? These strains are visually indistinguishable from each other. They look exactly the same. In fact, when we sequence certain marker genes, we can't differentiate them based on sort of fingerprint marker genes. They're very closely related to each other. But the difference between these strains, the toxic version and the non-toxic version, is the presence of these genes called the MCY genes. These are the genes for the biosynthesis of the toxin. Okay? So some strains have the genes to produce the toxin, and other strains do not. And the question is, why? Right? What controls the presence of these genes and therefore the production of the toxin? Well, there's been decades of active research onto this that have led to a couple of different hypotheses. One is that temperature affects toxicity. These toxic strains seem to like it hot. Okay? They are favored under warmer conditions. Number two is the concentration and form of nutrients. I told you that fertilizers are coming in. That's in the form of nitrogen and phosphorus. Even for nitrogen, there are different types of nitrogen. There's nitrate and there's urea. And it seems that the toxic strains here prefer high nitrate conditions. Okay, so there's some differences between these strains. And then finally, a very new theory is that the reason that these organisms produce this toxin is to protect themselves from certain environmental chemicals that are called reactive oxygen species. These are things like hydrogen peroxide that are naturally produced and that are deadly to these cyanobacteria. And so there's some evidence that the toxin, it's not there to make people sick or even to make other organisms sick. It's there to protect these organisms from hydrogen peroxide. Okay, so those are some of the potential explanations. So we've been studying this now in Lake Erie. So here's a schematic of what makes these cyanobacterial blooms tick, all right? So this is showing that, again, here are those rivers that I mentioned. These are the vehicles, the mode of delivering nutrients from the land into the lake. Okay, so this is bringing nitrogen and phosphorus that's run off of the land into the rivers. I told you that certain forms of the nutrients can favor these toxic forms over the non-toxic forms. So that's one possibility. Another possibility is that it's these reactive oxygen species. That's the hydrogen peroxide. And again, that also favors toxic versus non-toxic types. There's also some evidence that ecological interactions can affect toxic versus non-toxic types. For example, grazers, organisms that eat these cyanobacteria, might have preferences for toxic versus non-toxic types. Okay? So there are food web interactions happening over here that can affect these toxic versus non-toxic blooms. And then finally, we've also become interested in, okay, what produces these reactive oxygen species? And that turns out to be interactions between sunlight and dissolved organic carbon that is in the water here. And so think of dissolved organic carbon as the brown material that washes out of soils. Okay, so that's also related to runoff. My main point here is that this is complicated. Right? This involves not just biology, not just limnology. It involves runoff of land, water from land. It involves land use issues of how our farmers are plowing their fields. It involves what types of nutrients they're applying to their crops. It involves environmental chemistry. And to try and solve this complex problem, we have brought a team of researchers together here at the University of Michigan. They are highlighted here by color code, and that color code shows which areas of this system they're studying. Okay, so I'm studying the cyanobacteria. My colleague Rose Corey is studying these reactive oxygen species and their interaction with dissolved organic carbon and sunlight. We have colleagues in biology that are studying the other bacteria and the viruses that attack these cyanobacteria. Okay, so the point here is that this is a team effort between many different units at the University of Michigan, as well as our colleagues down at the NOAA Great Lakes Environmental Research Lab. Okay, so we're all working together, bringing many different disciplines together to study this complex issue. And in the last few minutes here, I just want to highlight some of our findings. So we go out to Lake Erie every week between May and November. We've just wrapped up our field season for 2015. And we sample 6, now we're up to actually 8 different stations in the western basin of Lake Erie. And I'm going to show you data today from this one station right here, Western Erie Station 12. This is right at that Toledo drinking water intake. Okay, and I'm going to show you this from 2014. This sampling is conducted by a really dedicated team of students and staff here at the University of Michigan. They go out every Monday under what are sometimes harrowing conditions, and in Lake Erie it can get quite rough. And they take samples that we then bring back to the laboratory to do these methods like extracting DNA and sequencing that DNA to understand which organisms are present through time. Okay, so I'm going to show you now, again, this is brand new unpublished data that I'm letting you in on. This is data from 2014, the Lake Erie microcystis bloom in 2014. In the top panel, I'm showing you concentrations of a pigment called phycocyanin. This is a pigment that's in most cyanobacteria, and we can take this as a proxy for the abundance of total cyanobacteria in these samples. Okay? So this shows you quite effectively when this bloom started. This bloom started in 2014 towards the middle or end of July. Okay, it really took off here at the beginning of August. And August 4th was a Saturday. I was actually driving through the city of Toledo when the drinking water was shut down. Okay, so this was the event. We sampled that, actually I think it was August 2nd was the Saturday. We sampled it that Monday, August 4th. Okay? So you can see that that bloom was sustained all the way to the end of October, or at least the middle of October here, at least in terms of abundance. There were some ups and downs, but this bloom basically happens throughout the year. Now in the lower panel, I'm showing you the concentration of this toxin, microcystin, all right? And what I want to point out is that at first, early in the season, that really tracks the abundance of the cyanobacteria. Do you see how it's really well correlated with cyanobacteria? But as we get towards the later part of the season here, the toxicity really drops off. Okay, so we go from a highly toxic bloom early in the season to a low toxicity bloom later in the season. And you might think, well, you're looking at total cyanobacteria here. Maybe it's just that the toxin-producing cyanobacteria were not there, but there were other cyanobacteria present. So to get at that question now, I'm showing you the relative abundance of different cyanobacteria. They're color-coded for different groups. And I just want you to focus on this green one here. This is Microcystis aeruginosa. This is the culprit for the toxin production. And the main point is that this one species essentially dominates this bloom throughout the whole year, from July until October. And even in these low toxicity phases here in October, it's still that same species of Microcystis aeruginosa. So it's not a different species that's causing this low toxicity bloom. However, you might recall that I said that some strains of Microcystis do not produce the toxin. And so our hypothesis was maybe we're seeing a shift in the strains of this bacterium that are present. So to get after that, we revisited our DNA sequencing techniques here. And we can tell you not only what is the total cyanobacterial population, but which fraction of those cyanobacteria are Microcystis. And then we can also directly sequence and assess the abundance of these MCY genes for toxin production. Okay, so we can quantify the portion of the Microcystis community that is capable of producing those toxins. All right, this is the ratio of MCY genes to the total Microcystis, which gives us the percent toxic Microcystis. So here now I've added on the bottom panel those results. This is now the percent toxic Microcystis through time. And what I want to point out is that indeed, in this early, highly toxic phase of this bloom, we see that the Microcystis community is dominated by organisms that can produce that toxin. They have these MCY genes for toxin production. As we go through the season, the proportion of Microcystis that can produce that toxin declines to essentially zero at the end of the season. So indeed, we are seeing a shift in the strains of Microcystis aeruginosa that are present. We never would have detected this if we were just using a microscope. Okay, these strains look identical. So these new DNA sequencing technologies are absolutely critical to our science here. So now, of course, the big question is why, right? Why do we see this shift in strains? Are there explanations that we could exploit in order to try and shift this balance earlier in the season? And I don't have answers for you yet, but I have some correlations. Okay? We've looked at many different environmental parameters, and there are two that very well match up with this change in toxicity that I've introduced you to. One is that there's a big shift in the availability of nitrate through the season. Okay, this red curve here shows the concentration of nitrate, one of those nutrients that's being washed into the lake. And you can see that there's a big decline in nitrate concentrations that matches up pretty well with this decline in toxin production. All right? This is of great interest to us because remember that I said that high nitrate favors these toxin-producing microcystis. Okay, so we're now doing experiments to verify this. The other possibility here that's also consistent with this pattern is that we have very high concentrations of naturally produced hydrogen peroxide early in the season. Again, this hydrogen peroxide has been shown to favor the toxin-producing microcystis. So we have two different environmental parameters which may very well be selecting for these toxin-producing microcystis. So now our efforts are directed in understanding these distributions, these patterns of environmental chemistry, and lining that up with our understanding of the biology of these organisms. And the end goal is that we may be able to come up with answers that inform policies, such as which fertilizers are most favorable or unfavorable to these toxin-producing cyanobacteria. Okay, just to summarize this second part, I've told you that this culprit, this Microcystis aeruginosa, can occur in a couple of different varieties. It can either produce the toxin or it may not produce the toxin, depending on the strain. I've showed you that there is a systematic shift in the toxin-producing form to the non-toxin-producing strain through the season, and that this correlates very well with certain parameters of environmental chemistry. Okay? This suggests to us that these strains are ecologically distinct from each other, right? They are biologically differentiated, and if we can understand their biology, we might be able to understand the toxicity in Lake Erie. So to get after that, we're now analyzing organisms that we've cultured from Lake Erie. We're also analyzing those environmental genomes and transcriptomes that I introduced you to across this big shift in toxicity and chemistry. Okay, so in the last minute or two here, I'd like to leave you with one thought and theme to tie all of this together, and that is time. I started today by talking about the oxygenation of Earth over billion-year timescales, and I showed you that in order to understand this pattern, we need to study microorganisms over hourly timescales. We also talked about Lake Erie and the seasonal transition from a highly toxic bloom early in the season to a non-toxic bloom late in the season. This highlights to us that we can't just study Lake Erie in July or August. We need to study it throughout. We need to study these microbial systems on a timeline that's relevant to them. Okay? So with that, I'd like to thank you for your time, and I would like to acknowledge some of the many people that have been involved in these projects. It's been funded by the National Science Foundation, by a fantastic program that we have here to support associate professors. This has allowed us to buy those microsensors that I told you about. There's a large number of colleagues and students and postdocs and staff that are involved in both of these projects. These are both very much team-oriented projects. And finally, for the Lake Erie project, that's funded by the University of Michigan Water Center. So with that, I'd like to thank you for your time, and if we have time, I'd be happy to answer any questions. Thanks.